Is aging the common fate of all life on Earth? Or is it a side effect specific to certain organisms? This is one of the oldest questions humanity has ever asked. Since ancient Greece, philosophers, alchemists in the Middle Ages, and biologists and physicists in the modern age have grappled with the same problem. Why do we age? And is it possible to change this process? The answer to this question should primarily be sought by looking at nature. Nature does not offer a single model of aging. Aging is not uniform in nature. Let's consider a small mouse. Its maximum lifespan is approximately 2 to 3 years. Fast metabolism, constant stress, and predator pressure. For these creatures, longevity is not an evolutionary advantage. Now let's look at a squirrel. It can climb trees, hide, and is agile and intelligent. These characteristics keep it alive, and it can live for 20 to 25 years. The situation changes when we move on to primates. Most primate species live for about 50 to 60 years. The longest recorded lifespan among chimpanzees belongs to Anne the Little at 77 years. But what about humans? The longest documented lifespan in humans is 122 years. Jean Calment not only observed this, but also provided evidence. However, it would be a big mistake to stop there, because humans don't hold the record among mammals. Those who don't age and those who live very long. Some whale species can live up to 200 years. Furthermore, these creatures show no signs of aging in the sense we understand it. Even more interesting, some creatures, like Hydra, show almost no signs of aging. These organisms maintain such a balance in cell renewal that it's even debatable whether they are biologically aging at all. This picture tells us something very clear. Aging is universal, but it is not inevitable, and it's certainly not uniform. But what about humans? Fate or a script? Now we come to the most critical question. Is the rate of human aging determined at birth, or can it be changed throughout life? The answer to this question lies in genetically identical individuals, in identical twins. Long-term twin studies conducted in the 2000s revealed a striking fact. Genetically identical people are exposed to different sunlight conditions, have different smoking habits, experience different weight fluctuations, and age at different rates. In other words, genes provide a framework, but the environment and lifestyle complete the details. Aging is not a matter of time. It's a matter of environment. One of the most striking examples demonstrating this is space. Astronauts who spend months on the International Space Station show signs of aging much faster compared to people on Earth. According to NASA reports, muscle loss, sarcopenia, bone loss, osteoporosis, increased cardiovascular risk, vision impairments, kidney dysfunction. Many of these effects are diseases normally associated with aging. However, these people didn't age. They remained the same age, only the environment changed. Why does space age us? Because space is an extreme stress test for human biology. Things we don't notice on Earth become vital in space. For example, no gravity. Fluids in the body distribute differently. Blood pressure, regulation, is disrupted. Bones weaken because they don't carry weight. Muscles atrophy because they are not used. Even when astronauts sleep, they have to place a fan next to their faces because carbon dioxide doesn't disperse on its own like it does on Earth. Add to this cosmic radiation, the Earth's magnetic field protects us. However, in space, especially on Mars missions, this shield is absent. Cells experience more DNA damage, repair systems are strained, aging accelerates. What is aging actually? Here we come to a very important point. Aging is not simply the passage of time. Aging is a decrease in the ability of cells to repair damage, a deterioration in energy production, a decrease in the capacity to maintain balance. According to modern biology, aging is the gradual weakening of an organism's ability to protect itself. This perspective is shared by many scientists, from Nobel Prize-winning biologists to modern aging theorists. So what is the conclusion? We can now clearly state, aging is a universal feature of life. However, it does not operate in the same way in every living thing. It is not a genetic destiny. It can be accelerated or slowed down by the environment and conditions. Aging is not a fixed fate, but a dynamic process. And even if people can't stop this process entirely, they can significantly influence its course. The kidneys may not be the most stress-sensitive organ, but they are one of the quietest testing grounds of the human body. That's why scientists gave a study on Mars Journey's a striking name: cosmic kidneys. This study asks a simple but unsettling question. If a person leaves Earth for a months-long journey to Mars, will they develop kidney failure before arriving? Will they need dialysis or a transplant? This question isn't just about space. It's about what aging is. Is aging universal? An inevitable price of life? For a long time, aging was thought to be an inevitable prerequisite of life. We are born, we grow, we age, and we die. But when we look at nature, this story begins to break down. Some things age rapidly, some age very slowly, and some hardly age at all. This fact tells us aging may be necessary for life, but it is not homogeneous. Aging is not a preserved, unchanging biological program. In other words, the aging process is reversible. So, what exactly is aging? Aging cannot be treated as an abstract philosophical concept. To understand it, one must look at the concrete, visible changes. Aging is the sum of tangible changes that occur in the later stages of life. And these changes are generally grouped under two main headings. 1. Visible changes, seemingly innocent warnings. The first group consists of changes that are often underestimated: gray hair, wrinkled skin, decreased elasticity. These are often seen as cosmetic problems, but they are not. For example, skin. Skin is not just an aesthetic surface. It is the body's greatest defense barrier. With age, skin thins and its permeability increases. What does this mean? You become more vulnerable to infections. You become more vulnerable to toxins. You'll have more difficulty maintaining heat and fluid balance. In other words, even seemingly harmless changes indicate a decrease in the system's protective capacity. But this isn't the truly frightening face of aging. 2. Decline, the silent dissolution of health. The real dark side of aging is the loss of function. At this point, the picture changes. Heart attack, stroke, cancer, Parkinson's, Alzheimer's. These are no longer just signs of aging, but age-related diseases that lead to the death of many of us. For a long time, these diseases were considered inevitable. That's just how old age is, they said. But science is now questioning this assumption. Why are age-related diseases different? So, what distinguishes age-related diseases from those seen in young people? Why are diseases like malaria, influenza, or tuberculosis treated differently? There are two key differences here. 1. They are multifactorial. Age-related illnesses usually don't have a single cause. Take kidney disease, for example. Causes may include toxic effects of certain medications, high or irregular blood pressure, chronic inflammation, oxygen and nutrient deficiency, metabolic imbalances, So there isn't just one enemy. Multiple small attacks eventually overwhelm the system. That's why doctors give you long lists. Don't smoke, stay away from sugar, reduce salt intake, exercise, manage stress, because the risk isn't in one place, it's everywhere. 2. Multiple diseases, multiple conditions. Another common occurrence with aging is the simultaneous onset of multiple diseases. In a person with heart disease, diabetes, kidney failure, atherosclerosis, all can occur at the same time. This raises the question, which caused which? The answer is usually, they are all different aspects of the same fundamental deterioration. Aging is a process, not a disease. Here we come to a critical perspective. Aging itself is not a disease. However, it is the ground that makes diseases possible. Modern biology defines aging as the organism's loss of its capacity to maintain its own balance over time. This definition is very important because it associates aging with not chronological age, but cellular and systemic resistance. Why are space kidneys so important? Mars missions show us that aging can accelerate under extreme conditions. In space, there is no gravity. Fluid balance is disrupted. Cells are exposed to more radiation. Organs wear out faster than normal. So aging isn't just about time. Environment, load, and pressure are also part of aging. This means the rate of aging is not entirely constant. Consider a small mouse. Its maximum lifespan is about 3 years. Squirrels living in the same ecosystem, able to climb trees and escape predators, can live up to 20 to 25 years. When we look at primates, this period extends to 50 to 60 years. The longest known lifespan in chimpanzees is 77 years. In humans, Jean Calment pushed this limit by living 122 years. But here's the interesting thing. We don't hold the record among mammals. Some whale species can live up to 200 years. Even more striking is that some creatures, like the hydra, show no signs of aging. Time passes in these creatures, but no biological decline is observed. This tells us something very clearly. Aging is not a universal and immutable precondition of life. It is not a fixed program maintained across species. So what is aging then? We cannot treat aging as a mystical or abstract concept. Aging is a concrete and measurable biological change. It manifests itself on 2 main levels. First, there are visible changes: graying of hair, wrinkling of the skin, loss of elasticity in tissues. These usually do not immediately threaten life, but they indicate a weakening of the body's protective capacity. The second and most critical aspect is functional decline. Cardiovascular diseases, stroke, cancer, Alzheimer's, Parkinson's. These become more common with age and often result in death. Therefore, for many years, aging was considered an inevitable decline. But science didn't stop there. Studies on genetically identical twins clearly showed that the rate of aging isn't determined solely by genetics. A comprehensive study published in 2009 revealed that environmental factors have a dramatic effect on aging. Even seemingly simple factors like sun exposure, smoking, and lifetime weight fluctuations can significantly alter the rate of biological aging. This means the course of aging can be altered. One of the most striking examples of this idea is space research. Studies on astronauts who spent months on the International Space Station showed that microgravity and cosmic radiation have effects on the body similar to those of aging. Muscle loss, osteoporosis, vision problems, cardiovascular risks. Many of these are problems we normally see in old age. But space emerges as an extreme environment that accelerates this process. The kidneys, in particular, are extremely sensitive to these stresses. A study titled Cosmic Kidneys questions whether astronauts undertaking long-duration journeys to Mars will face a risk of kidney failure. In other words, could a person need dialysis or a transplant before reaching their goal? This question alone says a lot. Aging is a biological response to environmental stressors. So why are age-related diseases so diverse? Diseases like malaria, influenza, or tuberculosis usually have a single cause, a pathogen. Therefore, they are relatively easy to target and treat. However, age-related diseases are multifactorial. A high-fat diet, smoking, inflammation, oxygen deficiency, drug side effects, all can combine to cause the same disease. This is why multiple diseases emerge as we age. More than one disease develops simultaneously, and it's not clear which is the cause and which is the effect. Looking back to the early 1900s, most people died from infectious diseases. Medicine has largely brought these diseases under control. But the price is different. People are living longer, and age-related diseases are becoming more common. The more than 200% increase in kidney disease in the last 30 years is one example. In the US alone, the cost of these diseases to the healthcare system amounts to hundreds of billions of dollars annually. The situation is no different in Europe. And what's truly frightening is this. By the time we reach 50, half of people are living with at least one age-related disease. By the time we reach 60, that number approaches 2. The situation worsens exponentially with age. So the big question is, why don't we have drugs that target aging? To date, no major regulatory body has approved a drug that directly targets aging. There are 2 main reasons for this. First, we don't fully understand the mechanism of aging. The causal chains are still incomplete. Second, the duration and cost of clinical trials are incredibly high. It takes decades to show that you're slowing down aging. Furthermore, modern medicine is still accustomed to addressing diseases individually. Yet aging is a holistic process with no single target. And perhaps now we should ask, can we treat aging not as an enemy, but as a manageable biological process? Science stands precisely at this threshold today. When you look at the phenomenon of multiple diseases emerging, you clearly see that these are not random, independent problems. Rather, they are different aspects of a complex network interconnected by invisible links. Heart disease, kidney failure, diabetes, neurological disorders. These often appear in the same body, within the same time frame. This gives us a very clear message. There is no simple problem. There is a complex systemic problem. This is precisely where the classical medical approach falls short. Because when you open any medical textbook, you see that diseases are dealt with separately. Cardiovascular diseases in one section, kidney diseases in another, and neurological diseases in yet another. This approach works for diseases with a single cause. However, in a multifactorial, multi-layered process, like aging, trying to understand the picture by breaking it down into parts causes us to miss the whole. So, if we can't use the standard approach, where should we look? Here, we should take a step back and consider, in today's society, what should we do when faced with a complex and multidimensional problem? The answer is usually the same. We turn to computational systems, big data, modeling, and of course, artificial intelligence. But can AI really intervene in the aging and multi-disease spiral? There's already a very strong example showing it's possible. AlphaFold, developed by Google, This AI system can predict the three-dimensional structure of proteins simply by looking at their amino acid sequences. This was a revolutionary step for biology, because if you know what a protein looks like, you also know how you can influence it. In terms of drug development, this is like going from navigating in the dark to navigating with a map. Why was AlphaFold so successful? Because it had incredibly rich data sets. Thousands of protein structures obtained over decades using rigorous techniques like X-ray, crystallography, and NMR. AI was able to learn from this past information and generate predictions about the future. However, the situation is very different when it comes to aging. Because there is no clear target here, there is no single structure that we can call the molecule that initiates aging. More importantly, we don't yet have the comprehensive datasets necessary to discover new targets. Therefore, if you unleash AI directly into this field, it becomes an explorer without a compass. It makes predictions, but doesn't know why it's doing them. Does this mean AI is useless? No. But it means it won't be sufficient on its own. The critical point here is that we need a framework of understanding in which AI can be placed. We can explain this with a simple analogy. If you give AI only a chessboard and a few pieces, it's unrealistic to expect it to win. But if you give it all the rules of the game, you can try it even against a grandmaster. So the question is, what kind of framework do we have to understand aging? One of the most commonly used frameworks today is the approach known as signs of aging. This approach categorizes biological changes that occur later in life. Mitochondrial dysfunction, genomic instability, cellular senescence, that is, the accumulation of senescent cells. These are often called zombie cells because they don't die, but also don't function and cause harm to their environment. This list is incredibly valuable for defining aging. However, it has a major shortcoming. It doesn't provide a clear answer to the question of why. Why do mitochondria deteriorate? Why does DNA become unstable? Why do cells become zombies? And more importantly, how do these microscopic changes translate into macroscopic consequences? Wrinkles, muscle wasting, organ failure, and cognitive decline. Here, we need to broaden our perspective. Let's think of biology not as a machine made up of individual parts, but as a house. This house has a foundation, walls, an electrical system, and plumbing. If you repair a crack in the wall, but the problem in the foundation persists, the house will eventually be damaged again. Aging works exactly like that. The problem isn't in a single room, but in the entire structure. And perhaps the way to truly understand aging is not to chase after individual diseases, but to understand the fundamental balance that sustains the system, namely biological integrity. Once a system is established, it wears down over time. This is true in architecture as well as engineering. Parts wear down, micro-damage accumulates, and eventually the structure loses its function. Aging has long been explained from this perspective. Life is established, then damage accumulates, and the inevitable end comes. However, while this explanation seems logical at first glance, a deeper examination reveals a significant gap. This is precisely the point raised by a renowned evolutionary biologist in the 1950s. He argued, if nature can build an extremely complex, organized, and functional organism from a single cell with almost perfect precision, why is it so unsuccessful at maintaining that system? In other words, if the real miracle is not the creation of the organism, but the inability to maintain what has been created, then there is a much deeper problem to address. This perspective shows that viewing aging solely as accumulated damage is insufficient, because damage can be a result, but it is not the sole cause. Biology does not function like a malfunctioning machine. Rather, it is an active system, constantly regulating itself and striving to maintain balance. Therefore, we need to reformulate the question, why doesn't the system protect itself as it used to? At this point, attention turned to genes. Perhaps aging is a genetically programmed process. The Human Genome Project was born with this hope. The goal was to map all the genes in human DNA and thus solve the root causes of big questions like aging, disease, and death. When genome sequencing was completed, expectations were high. However, the results did not meet these expectations. Large-scale studies showed that the influence of genetics on aging and premature death is far more limited than we thought. In analyses based on data from hundreds of thousands of people, only a very small fraction of individual differences in aging and major diseases could be explained by genetic predisposition. In contrast, the influence of environmental factors was much greater. Living conditions, stressors, habits, and environmental burdens appeared to be far more decisive than genetics. This finding opened a critical door. Genes are not destiny. Genes offer potential, but it is the environment that determines how that potential is realized. Therefore, understanding only the genome is no longer enough. The real issue is understanding the conditions that genes encounter throughout life and how they respond to those conditions. This is where a new approach has emerged, the Human Exposure Project. This approach focuses not just on genes, but on everything a person is exposed to throughout their life. Air, water, nutrition, stress, social conditions, chemicals, light, gravity, even the city they live in. It tries to understand how all of these interact with genes. This is a far more complex task than simply reading the genome, because here, there is not a static text, but a constantly changing flow of life. Two people with the same genetic makeup can be biologically directed in completely different directions when they live in completely different environmental conditions. Aging is precisely the long-term result of these interactions. Therefore, it is misleading to view aging as the result of a single gene, a single injury, or a single mechanism. Aging is the disruption of the balance in the organism's relationship with the environment over time. The system is still functioning, but it can no longer adjust itself with the same precision. That is why many research groups are focusing on this area. Because understanding aging means understanding how life is sustained. And the answer to this question lies not just in the genes or just in the environment, but in the constant, dynamic, and fragile interaction between the two. Now, the real question is, if aging is not a program and not just damage, what is the fundamental plan that keeps the organism afloat? And when this plan breaks down, why does the entire system begin to collapse together? That's precisely the next step. If you want to understand biology, you must first know what we are and how we work. Because to unravel the processes that kill us, you first need to understand the system. And the most fundamental concept here is this: we are not just our genes. We are a dynamic, interactive system. Let's do a thought experiment to illustrate this. Close your eyes and imagine a single bacterial cell. Now, put this cell in a jar filled with glucose as an energy source. What happens? The cell rearranges all its proteins to make the best use of the glucose, changing its structure and function. Now put the same cell in a different jar, this time with maltose as an energy source. Again, all the proteins change, this time to use the maltose most efficiently. The DNA is exactly the same. The genes haven't changed, but the cell has completely changed its appearance and function. The lesson here is very clear. Biology is a system, a network of interactions. The system determines which genes will be active and which will be deactivated. Therefore, the structure and function of an organism are shaped not only by its genetic code, but also by how those genes are used. Every system has risks: computers, machines, computing systems. The fundamental risk in all of them is limitation. You cannot optimize all functions within a system simultaneously. When you make a choice, you have to compromise on another function. In biology, these trade-offs are reflected in pathological pathways. You can think of pathological pathways as short circuits in a system. When the normal interaction between molecules occurs in the wrong place or at the wrong time, unwanted effects occur in cells and tissues. These molecular-level errors manifest as structural and functional disruptions at the cellular level. Therefore, biology is not simply a matter of a single molecule or gene error. Rather, it is a complex network of all interactions within a system. At the root of aging lies the accumulation of small disruptions and short circuits in this complex network. The changes seen at the cell, tissue, and organ level are the result of these pathological pathways, which begin at the molecular level and interact with the entire system. From this perspective, viewing aging simply as damage accumulation or genetic programming is insufficient. The key is to understand that an organism is a dynamically functioning system, and that this system is in constant equilibrium and trade-off at every level. This is where we must begin to understand biology. You cannot solve aging and disease without understanding the system. And the complexity of this system both complicates and directs our search for answers. The point where cellular balance breaks down, the silent beginning of disease. The human body is an incredibly complex system where billions of cells work together in perfect harmony. In a healthy organism, cells multiply neither too fast nor too slow. They are neither excessively large nor excessively small. But when this delicate balance is disrupted, a process that often goes unnoticed emerges: dysfunction of cell division. Some cells divide more than necessary. In medicine, this is called hyperplasia. Others show insufficient cellular adaptation. This can lead to hypoplasia, or, over time, atrophy. When cells become larger than normal, this is called hypertrophy. All these concepts point to the same fundamental problem: cells getting out of control. Even more striking is when a cell transforms into an entirely different type. This process is called pathological transdifferentiation. That is, the cell abandons its own existence and takes on a hidden role within the body. These events alone may not always create a major problem, but when this type of disruption becomes widespread, loss of function becomes inevitable. This is where what most of us call age-related deterioration comes into play. The invisible path, from molecule to disease. One of the most powerful aspects of understanding this process is that it connects us directly to a framework that extends from the authoritative level to what is clinically presented. The signs of aging lie precisely between these two levels, the gray area between deterioration and failure. For example, osteoporosis, or bone loss is a condition that people face. Our bones are not static structures. They are constantly undergoing a process of destruction and reconstruction. One of the structures managed in this process is osteoclasts. Under normal circumstances, osteoclasts break down old or damaged parts in a controlled manner. However, in this process, the balance is disrupted and complementary connections become active. As a result, the foam of osteoclasts begins to break down faster than it should. Bone density decreases, the structure weakens, and the risk of fracture increases significantly. Factors that trigger this process include hormonal changes, especially drops in levels, and chronic discharge. A similar vascular mechanism system is also observed: the silent transformation of vessels. Long-term monitoring of high blood lipid levels, glucose regime, and flow can activate critical circuits in the smooth muscle space of vessels. These are normally responsible for protecting the vessels. However, problematic changes occur, and this new structure gradually begins to transform into something else that more closely resembles bone. As a result of this transformation, calcification occurs in the vessel walls. The vessels harden, narrow, and the associated risk increases significantly. Some surgeons even report hearing a cracking sound in the vessels of elderly patients during surgery. This is not an abstract analogy. It is a concrete consequence of the condition. Why are these diseases multifactorial? A large proportion of age-related diseases cannot be reduced to a single cause. Many factors play a role simultaneously, including genetics, environment, nutrition, hormones, stress, and lifestyle. Could there be a simpler explanation for this complexity? Yes. If more than one different factor is linked in the same pathway, the result is most likely a multifactorial condition. In reality, there is only one breaking point at the beginning of the chain. When damage or dysfunction begins, it often creates a chain reaction. One pathological pathway triggers other problematic pathways. This brings us to a crucial point. Why do most people experience multiple diseases simultaneously as they age? The common root: universal pathological pathways. This comprehensive and powerful concept offers a universally diverse explanation. That is, once activated, the underlying dysfunction continues similarly in different organs and tissues. These pathways are in play: one disease in the heart, another in the vessels, and a completely different picture in the bones. From the outside, these appear as different transmissions, but deep down, they are different manifestations of the same biological process. This approach is extremely important because it offers a testable prediction. Good science does exactly that. It offers achievable and measurable predictions. The critical question: can these pathways be targeted? If these pathways are universal, is it possible to target them directly? And more importantly, could such an intervention alleviate multiple age-related diseases simultaneously? To illustrate this, consider a complex network. In this network, some nodes represent the failure of a large part of the system. Targeting the right node creates maximum impact. This idea is similar to the definition of a factor unit in the financial world. In a complex system, identifying and addressing the fundamental characteristics that cause the greatest harm can make the entire system more efficient. The situation is no different in biology. Conclusion: understanding the system, not the disease. This perspective teaches us that aging and reproduction are not simply the inevitable result of time. In most cases, They are the result of long-term failures in visible pathways. Real progress can be achieved not by trying to suppress individual diseases, but by understanding these common problem pathways and developing strategies to restore balance. Because a small but accurate intervention at the system level can have a much greater impact than we realize. Perhaps the real question is, how much more effective could the results have been if these pathways had been identified earlier? Maximum protection and the medicines of the future. So, how can we achieve maximum protective effect in preventing or reducing life-threatening diseases? A key clue lies in some of the secrets being uncovered. These are GLP-1 agonists, which are beneficial in treating obesity, Ozempic, semaglutide, and others. Initially developed for type 2 diabetes, these drugs were later rediscovered for the treatment of obesity. However, current research suggests that these programs may have a much broader potential for impact: neurodegeneration, osteoporosis, and a range of life-related diseases. Why? Because GLP-1 appears to target a regular pathological pathway. But there are also problems. First, the mechanisms are not yet fully understood. Second, GLP-1IC directly transmit genetic programs. These programs represent an evolutionary pattern selected over many years. Disruptions in this process can lead to side effects such as sarcopenia, loss of function, macular degeneration, or pancreatitis. At this point, the question arises, is it possible to develop a new group of drugs that mimic GLP-1s, but without the risk of side effects? The basic idea is this. Using artificial intelligence, we propose identifying a new node that has a systemic effect but directly modifies genetic programs. There are two standards for this: well-resolved mechanisms, accurately knowing which events trigger which processes; unprogrammed node points, selecting targets that are not genetically involved in evolutionary programming. This minimizes the risk of side effects. This idea may have scared you a little. Isn't playing with cell death dangerous? Does it increase the risk of cancer? You're right, because cell death is a critical part of normal biology. However, when the right node points are selected and the intervention is controlled, it may be possible to target aging and many fundamental details. In short, GLP-1s show us only the tip of the iceberg. By combining instantaneous, short-term intelligence and helpful biology, we can develop new drug therapies that can get to the root of relevant diseases and alleviate multiple diseases simultaneously. Cell death and aging, necrosis and opportunities. In biology, cell death occurs in two main ways: programmed, beneficial cell death. These are cells that are safely killed after DNA damage is observed. This process is critical for reducing cancer risk, promoting tissue regeneration, and facilitating wound healing. For example, a fracture requires this type of controlled cell death to clear the damaged bone tissue and prepare it for new bone grafting. Uncontrolled, harmful cell death, necrosis. This occurs through the bursting of cells and their dangerous proliferation into surrounding tissue. Necrosis initiates chains of degeneration, triggering the formation of senescent or zombie cells and leading to scar tissue or fibrosis formation through chronic, persistent aging. In short, many age-related oscillations occur. According to the conventional view, intervening in necrosis is nearly impossible. It is too chaotic and uncontrolled. However, with the widespread adoption and appropriate expansion of factor programming, it may be possible to disrupt this adverse cell death. If we can achieve this, it will be a revolutionary step in preventing age-related degenerations and multi-organ diseases. At this point, sensitive organs such as the kidneys are particularly important. For example, the term cosmic kidneys helps us understand the role the kidneys play in proliferation in relation to this type of data. Necrosis preventing drugs and kidneys, an accelerated aging model. The kidneys are one of the most stress-sensitive organs and therefore offer an ideal model for observing accelerated maturation processes. Various stressors, drug side effects, low blood pressure, oxygen and nutrient deficiencies, malnutrition, can lead to necrosis in the renal tubules. This can result in acute tubular necrosis and acute kidney transplantation. The stages of the necrosis chain are initial cell death, leading to chronic inflammation, senescent cells, and fibrosis. This, in turn, leads to the development of age-related chronic kidney disease. To date, there is an unmet need for treatment for kidney diseases where necrosis cannot be addressed. Current treatments are limited to dialysis and transplants. Studies at the molecular level have shown that intervention in necrosis may be possible. The key point is the simultaneous deactivation of the specified calcium channels. Calcium is the cell's critical signaling molecule. Disruption at these levels leads to the simultaneous binding of multiple signaling pathways under stress and cell death. Targeting this mechanism could potentially halt production system-wide. In this approach, along with laboratory experiments, an anti-necrosis agent tested in artificial human tissue allows 90% of cells that would otherwise die under extreme stress to survive. Furthermore, central necrotic nuclei were inhibited for up to a week in the tissue engineering model. These data demonstrate that anti-necrosis agents could be incorporated into clinical trials in an accelerated aging model of the kidney. The aim is to prevent age-related changes and provide a systemic treatment within just a few years. The potential of the drug extends beyond kidney function. It could also benefit extreme applications such as space exploration, a program supported by NASA and the UK Space Agency.